So now we'll finish by circling back to the broad goals and applications of single-cell RNA-seq technologies, and basically just do a little bit more visualization on what those goals are.
Right, and so one of the next major applications of single-cell RNA sequencing is obviously visualization of your data, which essentially amounts to plotting your samples or your cells that you've sequenced in a 2D scatter plot. And so here's a classic, what's called a t-SNE visualization of a single-cell RNA-seq dataset. And so in your typical visualization, you are visualizing a large dataset of single-cell RNA-seq, where the x and the y-axis are just kind of a 2D representation of your cells, and each point represents a single cell in your dataset. And in this particular case, a method called t-SNE is used to visualize your data. Other common software includes things like UMAP. These methods like t-SNE and UMAP tend to do a better job visualizing data compared to PCA because they're what's called nonlinear dimensionality reduction techniques. But they're less interpretable in the sense that we don't actually know what these individual axes mean. And so basically one of the points of visualizing your data are to basically see how cells cluster in your data set. So here again, I mentioned that each point represents an individual cell. And one of the first striking features about this visualization is that you can see that points are not randomly distributed around this figure. Points are grouped into different clusters. And those clusters seem to correspond to the tissue or cell type of origin from which they were sequenced. And so basically visualization gives you a kind of first pass look at your data and allows you to kind of roughly guess how many kind of cell types are in your data set, where you might tentatively think that each grouping of cells might correspond to a putative cell type, because grouping of cells group together when they exhibit similar expression profiles. And so from this visualization is essentially like a starting point from which you can, for example, identify candidate cell types, and then identify, for example, cell markers or kind of signature genes of each cluster by taking each individual cluster and, for example, doing differential expression tests to try to figure out which set of genes are uniquely expressed in one cluster of cells compared to all other ones. And that in turn can then help you kind of try to identify what function that group of cells performs. Because if a particular cluster of cells only express, if they express one gene or set of genes uniquely and that are not expressed in any other clustering of cells, then that kind of tells you that that gene or set of genes are probably a unique function, represent a unique function of that cell or that group of cells. And so kind of through comparing different clusters of cells and trying to figure out which clusters express which cells, or which clusters express which genes, then you can kind of guess to see what kinds of cells you've sequenced in your data set. And so as I was just mentioning on the previous slide, after visualization, typically what you do is you perform a lot of cell type identification related tasks. And so you'd find clusters of cells corresponding to candidate cell types. You'd figure out, using differential gene expression analysis, what type of genes are uniquely expressed in one group of cells versus another, and then use those uniquely expressed cells as a way of trying to figure out what those cells represent. Something that we haven't really talked about is rare cell type identification. And so I briefly mentioned earlier in this lecture that one of the benefits of single cell analyses is that you can start to identify cells that are present in relatively small proportions in samples. And sometimes in the cases of tumor samples, for example, they might correspond to important types of cells like cancer stem cells. Some of the other important tasks include, sometimes it's important to characterize, for example, for a given sample, what proportion of that sample is composed of cells of one type versus another. And so sometimes this can be useful, for example, when you're studying immune cells sequenced from patients that are undergoing like an infection versus not. Because by looking at the changes in the types of immune cells that are present in the blood, then you can get some idea as to what type of immune reaction a given individual has. And so the final application we'll briefly touch on is, again, what I call trajectory inference. And so again, trajectory inference is really all about trying to characterize what changes in gene expression patterns happen as cells move along continuous processes like differentiation or reprogramming. And so again, trajectory inference is really about studying how cells decide cell fate. Right, and so here, the diagram on the right represents single cell data collected from cells that are differentiating from a pluripotent cell towards a myoblast. And basically you can see that with a high enough number of cells sequenced, you can see that when you visualize the cells in a 2D scatter plot, the cells kind of form a nonlinear line that starts at the red points and ends at the blue points. And so trajectory inference refers to a bunch of computational methods which are oftentimes applied to basically look at these 2D scatter plots and basically draw these so-called trajectories or differentiation pathways where the start of the trajectory, or what I've labeled here is time equals 0 for pseudotime, represents the start of, for example, differentiation, and the end of pseudotime, or where time equals n, is basically where the computational method thinks that differentiation basically has stopped. And so trajectory inference just refers to the huge number of methods available for finding these kind of trajectories and doing some downstream analysis to figure out what kinds of genes are changing in expression as you move from the start of differentiation to the termination of differentiation. And so it's worth pointing out that differentiation is obviously a complex process. And so in the simplest case, you can have so-called linear trajectories, where you only have essentially one starting point and one ending point. There can also be cases of, for example, what's called branch trajectories, where you start out with one kind of pluripotent cell, and you can basically, that cell can eventually differentiate into two different cell fates. And so it's a little bit more complicated to identify these trajectories, because you have to figure out where does the fork in the cell fate decision making happen. And so there's kind of different trajectory inference methods that can be applied to find these different kinds of trajectories. And so the final thing I want to say about trajectory inference is that because trajectory inference is one of the motivating applications of the single-cell RNA-seq protocols, there is a tremendous variety of trajectory inference software that you can use to infer trajectories. And so the last time that I checked, which is probably back in like February 2020, there were something on the order of like 130 different softwares that you can use to infer trajectories. And so it is insane how many different ways you can use to infer trajectories. And so it's basically impossible to kind of go over all of the different ways in which trajectory inference can be done, because there's just so many of them. But this figure here is really just to kind of illustrate to you how some of the more popular trajectory inference approaches work, because they can basically be boiled down to a few different choices that need to be made. Because most trajectory inference methods work by performing an initial step of dimensionality reduction to reduce noise in the data. And so you can use methods like principal components analysis, like we talked about, or basically like a million other ways of doing dimensionality reduction. And then after dimensionality reduction, then essentially you have to choose an approach to find these kind of lines or these trajectories through your data. And so because there's so many choices for performing dimensionality reduction, and there's so many choices for finding kind of lines in these visualizations, then that's why you can end up with like 130 different pieces of software to do effectively the same thing. And so again, something I want to mention is that we talked about single-cell RNA-seq in this lecture and mainly focused on that. But again, there's pretty much single cell based assays that have been developed for most of the assays that we've talked about in this course. And so again, there's kind of corresponding methods for doing methylation profiling at the single cell level, attack-seq, chip-seq, Hi-C, DAM-ID, and so on and so on. And so it's, there's just, this field is just moving at an incredible pace. And so just like for single cell RNA-seq, a lot of the methods like single cell attack-seq, Hi-C, and chip-seq, they suffer from a lot of these problems related to low input material. And so that's why these methodologies are currently still immature, in the sense that while you can technically perform the assays at single cell level, whether or not you can extract the same information out of those single cell assays is a whole other issue. And so in terms of summarizing the single cell RNA-seq portion of this lecture, basically the point that I've tried to make is that single cell biology and related assays reveal a lot of information that you can't detect from bulk data. Some of the main current applications of single cell RNA-seq are to study heterogeneity of bulk samples, look at the dynamics of transcription and gene expression, and identify new cells or do trajectory inference. From a practical standpoint, the data that you get from single cell experiments are still very much more noisy than compared to bulk assays, and they're much more difficult to analyze. And so the so-called software protocols for doing a lot of these downstream data analysis tasks are still kind of being worked out in the field. And a lot of the methods are still underdeveloped. Some things that I want to note that we specifically didn't cover in this lecture are, number one, the ever-growing set of single cell-based protocols that measure spatial transcriptomes. And so I said that for a lot of these single cell-based assays that we talked about, one of the first steps that you have to do is you have to dissociate cells from their complex tissues into single cell suspensions. And one of the problems with that is that you lose the original spatial positioning of the cells in, for example, tissues. And so for a lot of tissues, for example, like brain or liver, a lot of the functionality of a cell depends on its context. And so it depends a lot on what other cells are around it, and so on and so on. And so there's a lot of technologies now being developed where you can actually both do single cell sequencing, RNA sequencing, or at least, you can profile the expression level of a small number of genes and actually maintain their original position in the tissue. And so in that way, you can get both the expression profile and the position of cells in tissues, and so you get a lot more information out of that. We also didn't really discuss multimodal assays, and so multimodal assays refer to single cell sequencing protocols where you collect more than one type of data. And so, for example, site-seq refers to technologies that allow you to both measure RNA sequencing and certain protein measurements of the same cell. There's assays that allow you to do both attack-seq and RNA-seq at the same time for individual cells. Assays like patch-seq allow you to simultaneously measure RNA-seq of a neuron, measure electrophysiological recordings on those neurons, and measure the physical morphology of those neurons all at the same time. And so there's kind of growing interest in these multimodal assays because they allow you to connect both kind of like RNA and like attack-seq or cellular phenotypes on the same cells, and therefore kind of build models about how variation in different molecular phenotypes covary. We also didn't talk about a lot of the even the kind of more mechanical details of single cell RNA-seq. So for example, how differential gene expression tests kind of vary for single cell RNA-seq data compared to bulk, or even how quantification is dealt with. So even coming up with metrics for how to quantify gene expression is different for single cell RNA-seq compared to bulk RNA-seq, for example. But we didn't really touch on that in this lecture.